Hello students, welcome to EPG Pathshala. I am Anup Kumar Kapoor from Department of Anthropology, University of Delhi. Today we are going to talk on module Structural Polymorphism of Human Hair from the paper Forensic Anthropology. In this module, we shall be focusing on the importance of one of the most important evidence in personal identification, that is hair. The student will learn basic types of human hair like langoon, vellus, terminal hair, how they are formed and how they differentiate from animal hair and how should they be collected from different sites. Student will learn how to search for, collect and prepare them for further comparison and analysis in this module. Hair is a characteristic of mammalian skin. For some animals, it serves as a warm coating of thick hair to protect them from the harsh weather and hazards of environment, or to camouflage them as protection from attacks by their predators. Humans too have a fine layer of hair covering the entire body except palms and soles. Hair, which is composed primarily of the protein keratin, can be defined as slender outgrowths on the skin of mammals. Each species of animal possesses hair with characteristics length, color, shape, root appearance, and internal microscopic features that distinguish one animal from another. In human hair, found on the head, pubic regions, arms, legs, and body areas have characteristics that can determine their origin. On animals, hair types includes coarse outer hair or guard hair, the finer fur hair, tactical hair such as whiskers, and other hair that originate from the tail and mane of an animal. Components of hair. The hair is the major component of hair follicle, commonly examined microscopically in forensic hair comparison. It is a long, thin cylinder of keratinized cells and usually has three distinct cellular components. First is medulla, a central or a core portion running along the central axis. The second, cortex, the main components filling the inside of each hair shaft. And the third is cuticle, the main outer covering. Now I can give the example of a hair with a pencil. When you see a pencil, in the interiormost side is the lead, black color, then covered by the wood, that is the cortex, and outer structure when you see different coloration or different design, that is the cuticle. First is medulla. It is formed as a column of cells that produce a protein that is distinct from the proteins of cortex and cuticle, in that it contains the amino acids. Human hair is unique in that its medullary index, the fraction of the hair shaft's diameter that the medulla occupies, is very small, generally less than one third. Other species will have ratios much larger, usually at least half. The medulla's presence in human hair and the patterns of which differ from both person to person and from stand to stand on a single person's head. One hair may have absence of medulla, while another from the same person may have a complete or fragmented medulla. For this reason, medulla patterns are not very useful as forensic evidence and are typically only used for determining the species of the subject. The medulla contains large amount of mitochondrial as opposed to nuclear DNA. Mitochondrial DNA is passed from the mother to her offspring. As such, mitochondrial DNA may be of forensic value. Cortex. The cortex of the hair shaft is located between the hair cuticle and medulla and is the thickest hair layer. It also contains most of the hair's pigment, giving the hair its color. The pigment in the cortex is melanin, which is also found in skin. The distribution of this pigment varies from animal to animal and person to person. The cortex determines the color, flexibility, and tensile strength of hair strand. Tensile strength is the maximum amount of force the strand of hair can take in longitudinal direction without breaking. Cuticle. The hair cuticle is the outermost part of the hair shaft. It is a hard, single-like layer of overlapping cells. It is formed from dead cells which form scales that give the hair shaft strength and do the best job of providing protection for it. The hair cuticle is the first line of defense against all forms of damage. It acts as a protective barrier for the softer inner structure, including the medulla and cortex. The cuticle is responsible for much of the mechanical strength of the hair fiber. A healthy cuticle is more than just a protective layer, as the cuticle also represents the structure that controls the water content of the fiber. Much of the shine that makes healthy hair so attractive is due to the cuticle. Cuticles are often damaged by excessive mechanical manipulation such as brushing, heat, using curling irons, chemical processing, harmful ultraviolet sun rays or dust, and wind can cause wear and tear on hair and damage the hair cuticles. Also, cuticle cells may be elongate, tapering to a point, ovate, or most often flattened. There are three stages of human growth and replacement. These are as follows: anagen, catagen, and telogen. The hair follicle is a dynamic organ in which division, differentiation, and migration of cells occur in the various tissues of which it is composed. These processes give rise to the growth of hair fiber, which is formed as the result of the biosynthesis and hardening of the contents of medulla, cortex, and cuticle cells of the hair shaft. This growth, however, is not continuous process but one which ceases periodically and then starts again to be repeated in what is known as the hair growth cycle. The stage of this cycle in which hair grows actually is called anagen. It is when the follicle is full size and is maximally biochemically active. This is followed by a quiescent or regression phase called catagen, which metabolic activity and hair growth slows down and eventually stop at stage called telogen or resting phase. It becomes dead and falls off on its own. We see at the figure we found that is the anagen first stage active growth phase, that is 2 to 6 years. Then the second stage catagen, that is a transition phase, that is 1 to 2 weeks, and telogen, that is the resting phase, that is from 5 to 6 weeks. This shows the different stages of a hair from the childhood or the fresh hair to the old hair follows. Types of human hair. Head and body hair of humans is classified as intermediate hair combining the characteristics of bristle and wool hair. Four types of hair appear on the bodies of humans. The first one is primordial hair. It appears as early the third month of gestation, growing on the upper lip, the eyebrows, the palms, and soles of the fetus. They gradually disappear and are replaced by softer langur hair over the entire body. Second is the langur hair. It is a special downy hair that completely covers the human body inside the womb. It is like fur, but before the fetus is born, the langur hair is replaced by vellus hair. These are normally shed after the 6th month of gestation. They are fine, soft, unmodulated, and normally unpigmented hair. The surface of the langu hair is smooth with almost indiscriminate scales. It is replaced by vellus and terminal hair. Langu hair is often observed on an aborted fetus and can be useful in investigation of possible infanticide. Vellus hair. These are frequently short, fine, colorless hair found all over the body. Vellus hair helps to regulate body temperature. Vellus hair replaces languor hair on a human fetus at 36 to 40 weeks of gestation. The growth cycle of vellus hair is different from the growth cycle of terminal hair. At puberty, the androgen hormone causes much of the vellus hair to turn into terminal hair and stimulates the growth of new hair in the armpit and the pubic area. In men, this change in vellus hair occurs on the face and the body. Vellus hair grows individually or in small patches of varying or sometimes irregular length of 2 to 4 centimeters. These irregularities are governed by genetics and often persist throughout a lifetime. This kind of hair growth is usually not harmful. The lastly it is fourth is terminal hair. It is the type of natural hair to which most people refer in their everyday lives. It is the type of hair that grows on your head, in your pubic regions, and on almost all parts of your body. A hormone called androgen controls this body hair, also known as androgenic hair. There are three types of hair usually seen in animals. They are first vibrissae. They are the whiskers of many animals. They are normally tactile and sensitive, such as whiskers on a cat. Second is bristle. This is the coarse bristle that provides an animal with a protective coat. These guard hair can readily be identified by their distinctive appearance and morphology between various animal families. Third is the wool. Wool or fur provides insulation from wet and cold. These fine hair covers the bodies of all mammals. Hair analysis. Hair is a common type of evidence submitted to the trace section of analysis. On average, humans shed approximately 100 head hair per day. Because we are continuously shedding hair, forensic analysis of hair is beneficial to the establishment of association between individuals, a person, and an environment. For example, a questioned hair from a victim's gene is examined and found to be consistent with the suspect's hair. This examination has provided evidence that an association may have occurred between the suspect and the victim. A hair is a fibrous outgrowth from the skin of mammals and consists of three anatomical regions: the cuticle, medulla, and cortex. Characteristics within these regions are used to determine whether the hair is of human or animal, racial origin, or body area. Microscopic hair examination can also determine if a hair was forcibly removed, artificially treated, or diseased. A comparison microscope can be used to compare a questioned hair to a non-hair sample in order to determine if the hair are similar and if they could have come from a common source. A questioned hair and non-hair sample are viewed side by side using a comparison microscope in order to determine if they have similar characteristics. Color. The first step of hair analysis is to compare the hair color of questioned hair with that of suspect's hair samples. Hair color could be naturally different in two individuals of same race geographical area and sharing same gene pool, or sometimes differ in color when would have dyed or colored the hair. In either case, it brings out specific point of difference in forensic casework investigation. For this purpose, the forensic expert needs to compare minimum of 20 to 40 strands of hair samples obtained from scene of crime with that of procured from specimen. The expert matches the color of dried hair with the literature of SOP color card and ticks on the column of color that best matches hair color. He also needs to mention whether the hair were dyed previously or not. Once this preliminary investigation is done and reads positive results, he may further inspect the hair shaft microscopically for more detailed morphological analysis. Race. The hair evidence is most suited in case of sexual assaults, hit and run, murders, and child abuse cases. The expert needs to look for the presence of root of the hair strand. If it is absent or dead, it is a naturally found hair, whereas in the case of plucked hair, if the root is present and the bulbous, then DNA may be isolated. Once DNA is extracted, one can say about the sex of the owner of hair, race of the bearer, and if he or she suffering from any disease or poisoning also can be told. Examination of common hair features in wet mount slide. First, pull out, do not cut one strand of suspect's hair. Second, place the hair on a microscope slide. Third, add a drop of water and place a cover slip on top. Fourth, on high power, scan the entire length of the hair shaft, determine the type of medulla sketch, and label the features. Fifth, use the camera to take a picture of hair through a microscope lens. And lastly, find the root end. Make a sketch of what you see. Now this table shows racial characteristics of hair. Here we have identified three races: Negroid, Caucasoid, and Mongoloid, and then we have includes which includes in the case of Negroids of black color, Caucasoid, American, European, Mexican, Middle Eastern, and Mongoloid, Orientals, and American Indians. There are many other races, sub-races, but for your reference we have included only a few important ones. Then we see that in a table we have taken the diameter, cross-section, pigmentation, cuticle, and modulation. That is medulla. Now first example is the Negroid. The diameter varies from 60 to 90 micromillimeters. Cross-section is flat. Pigmentation is dense and clumped. Cuticle is absent. Medulla is prevalent. Then we take Caucasoid, which includes American, European, Mexican, Middle Eastern. The diameter varies from 70 to 100 micrometers. Then cross-section is oval. Pigmentation is evenly distributed and cuticle is medium, and modulation is uncommon. In the case of Mongoloid, the diameter varies from 90 to 120 micromillimeters. The cross section is round, pigmentation is dense auburn, then cuticle is thick, and modulation is never. So, from this source table, we can see that all the ethnic groups, here we have given the races, but all the ethnic groups or population groups, they vary in their characteristics of human hair. For species origin from hair, medullary index is predominantly studied along with the other tests for specific origin such as pigment distribution, scale patterns, etc. Medullary index is calculated as the diameter of the medulla divided by total diameter of the hair shaft cross section. If the value comes one-third or less, it is human hair, while if it is one half or more, it is a non-human hair. There are four different types of medulla in humans. They are continuous, fragmented, discrete, and absent. When we look at the diagram, four diagrams, the first represented by the continuous medulla, whereas the B there shows fragmented medulla, then C is discrete medulla, and in the case of D, medulla is absent. Examination of common hair features in a prepared slide. First, obtain a prepared slide of human hair along with two different animal hair slides. Second, view under the compound light microscope, first at lower power and then at higher powers. Choose a representative hair on each side. Third, scan the entire length of the hair. Determine the type of medulla if the hair is human, or medulla type and pattern if the hair is animal. The fourth, sketch the features of one representative hair from each slide. Include labels for the medulla, cortex, and cuticle. Your drawing should be large and detailed. Include the magnification used. Actual magnification number, not low, medium, or high. Examination of cuticle scale patterns. First, paint a thin coat of clear nail polish on glass slide. Cover an area about 1.5 cm into 2 cm. Second, gently embed the shaft of a hair into the nail polish. Leave the root and tip ends free of the polish. Third, before the polish has thoroughly dried, but after the surface becomes partially solidified, that is, after 1 minute, grab onto the root and tip end of the hair with two hands and lift the hair straight up to of the slide. You should now see an imprint of the hair in the polish. Fourthly, observe under microscope and make a sketch of the cuticle scale pattern. Use the camera to take a picture of the hair cuticle scale pattern through the microscope lens. Repeat steps 1 to 4 with one of the animal hair samples and make a sketch of the cuticle. Polymorphism. Polymorphism of hair was considered in terms of their origin in mammalian evolution, although forensic analysis of STR profiles is a well-established technique and has proved invaluable in linking individuals to crime scenes. Such profiles can give only limited information about the likely appearance of an individual. Therefore, in circumstances where there is no eyewitness evidence of a crime and a biological sample has been left at the crime scene by an unknown individual, it would obviously be of benefit to establish one indication of that person's physical characteristics, such as the color of the hair. Variation in human hair and skin color is caused by differences in the distribution, level, and type of the pigment melanin, which is produced in specialized cells known as melanocytes. Melanin exists as two distinct compounds, that is, eumelanin, which is brown or black in color, and phenylalanine, which is red or yellow, although human pigmentation shows a wide variety of phenotypes and is thought to be under the control of several genetic loci. Recent work has shown that in many cases the red hair phenotype can be associated with variants of a single gene, the melanocortin 1 receptor, that is MC1R, or melanocyte stimulating hormone receptor. Red hair contains higher level of pheomelanin than hair of other colors. Certain variants of MC1R have also been associated with different populations. In particular, it has been reported that one variant, that is ARG163 GLU, is more common in Chinese and Southeast Asian individuals than Caucasians, Africans, or Indians. It has been reported that red hair appears to be inherited in a recessive fashion in many families, requiring an individual to be homozygous for a given variant of the gene or compound heterozygous for two different variants in order for red hair to result. Verbasica significantly differ from hair by the structure. Apparently, they originated from methanoreceptors of Cytolothuria and developed only in the Thyrodontia, Cynodontia, and Panthotheria lineage, that is, Metatheria or Eutheria. No Vibrassa developed in the lineage of Thyrodontia, Cynodontia, or Prototheria, that is, Multituberculata, or Trichondonota, or Monotremata, where mechanoreceptors or granule structure of amphibian structures transformed into electro and mechanoreceptors. Hair originated in these lineages in parallel from keratin epidermis in correlation with the establishment of homothermy but independently of vibration. Wide and active adaptive radiation provided for a strong differentiation of hair, largely similar in different taxa. Spines originated by heterochrony of hair generations. Hair suitability for DNA examinations. Nuclear DNA analysis can be done on human hair. The trace section assists the DNA section by screening hair and determining their suitability of DNA testing. A microscope is used to examine the root end of the hair in order to determine if they are suitable. The hair in photo 1 is not suitable for nuclear DNA analysis, and in hair in photo 2 is suitable. If a hair is deemed not suitable for nuclear DNA analysis, that is, no root present or no tissue is attached, it can be sent for mitochondrial DNA testing. A root does not have to be present in order for a hair to be suitable for mitochondrial DNA testing. Structural polymorphism of human hair. Hair keratin associated proteins, known as KAP, are a major structural component of the hair fiber. In humans, 5 clusters of KAP genes have been identified, leading to the isolation of more than 80 individual KAP members. Of these, a cluster of high or ultra-high sulfur KAP has been characterized on chromosome 17 Q12 to 21. Within this cluster, however, there was an incompletely sequenced region in which the ultra-high sulfur, that is KAP4 genes were located. A recently reported human bacterial artificial chromosome clone, that is AC100808, finally covered the gap, which enabled us to characterize the complete set of KAP4 genes in this cluster. Analysis of the nucleotide sequence of AC100808 together with PCR amplification allowed us to identify numerous size polymorphisms in the KAP4 genes, which were mainly due to variations in the sequence encoding cytosine-rich repeat segments. Taken together, the data indicate the polymorphic allele of the KAP4 genes may have arisen through intragenic deletion and or duplication of sequences encoding the repeat structure during evolution. Here I would like to tell you 2 or 3 important points for your consideration. First is, usually when you identify hair at the scene of crime, you may get 1, 5 or 10. Normally it is suggested that more than 50 hairs should be present. In case 50 hairs not possible, then you can have 1 to 5 hairs for further analysis for its identification. In some cases, you don't get hair at the scene of crime, either on the clothes, on the shoulders, then you have to take the comb and go to the suspected criminal and ask him to comb his hair. When you comb your hair, some hair will fall down, and then you have to bring to the laboratory for further analysis. Again, when we talk about when you take the fresh hair, when you say when you see the structure of the hair, that is on the tip and the bottom of the hair, where you see the white structure, that is called the bulb. In case the bulb is dried, it means that is fallen on the floor and it is more than 1 or 2 days. When the bulb is fresh or wet, it means recently that has fallen from the head or different parts of the body. Now we can summarize the important points what we have studied in this module. Here can be important physical evidence at the crime scene. Hair normally falls from the body over the course of the day. It will stick to a number of materials, especially fabric or clothing. Hair is not easily destroyed even with exposure to moisture and decomposition of accompanying tissue. Thank you.